Hello, this is Jenny Frankovich. I will be working with the team today to talk about PANS and the current evidence for inflammation. So I'm Jenny Frankovich. I'm from the Department of Pediatrics and Immune Behavioral Health Clinic at Stanford University. Second will be Chris Pittenger, who will present on autoantibodies to cholinergic interneurons. And he is from Yale University. He's the director of the Yale OCD Research Clinic and Yale Program for Psychedelic Science, among many other positions. And then third will be Shama Raman, who is a postdoc in Betsy Mellon's lab, and they are studying inflammatory monocytes and brain homing repairing monocytes. And then last, we will have Tyler Prestwood present. He will be representing PJ Utz's work. Well, Tyler's leading the work in PJ Utz's lab on autoantibody profiles in PANS. And he'll also talk about his work to develop an assay to detect autoantibodies in G protein coupled receptors. And he is also a postdoc student and fourth year psychiatry resident at Stanford University. So my disclosures include funding from Neuroimmune Foundation, PANS Pandas Physician Network, Stanford Spark and other Stanford programs, Global Lyme Alliance, Dollinger Family Foundation, and other foundations. And this funding goes towards collecting samples, imaging research, and basic science research. So we started our PANS Pandas program about 10 years ago. And when we started, our goal was to look at all the different comorbid inflammatory diseases that co-occur with psychiatric symptoms, but the majority of patients coming to our clinic were wanting a workup for PANS and PANDAS. So ultimately, we developed a dedicated clinic to this program. As many of you know, PANS is a sudden, severe onset of OCD or eating restriction, and oftentimes the eating restriction it involves obsessive compulsive symptoms, including fears around contamination. In addition to the sudden onset of OCD or eating restriction, the patient must also have sudden onset of two other coexisting symptoms. So these are typically kids that have no underlying or very little underlying psychiatric problems and suddenly overnight develop very severe symptoms and severe disability. So as you can see, the top corner, I don't know what's happening here. Okay, we're going to skip that slide. For some reason, it won't stay on. But most of the patients in our clinic are relapsing and remitting, with a smaller proportion of patients developing chronic symptoms. There's been 4 imaging studies which suggest basal ganglia involvement, including the original study done in 2000 by Jay Geed. You can see in the far left panel where he had shown increased basal ganglia volume, especially in the caudate putamen and globus pallidus compared to controls. And these were new onset cases. And that study was followed by a study led by Dr. Kumar and Dr. Chugani, who looked at microglial activation in the basal ganglia using PET imaging studies. And he showed that there was increased uptake of this radio tracer that signifies microglial activation in both pandas and Tourette's, but pandas had more involvement. So the caudate and the globus pallidus, whereas the Tourette's was primarily the caudate. And then another study published in 2019, which indicated increased gray matter volume in the basal ganglia, whereas there was decreased white matter volume in the basal ganglia. And then the last study is a study led by Kristen Yam and Dr. Zhang at Stanford, and they showed increased medium diffusivity across all brain regions, but definitely the most prominent finding was in the basal ganglia. And there was more, that the finding of increased diffusivity was greater in patients that had these subtle findings suggesting chorea. but not overt chorea. So here's an example of a brain imaging study of a patient who got the MRI within 2 days of onset. That's very rare, or at 3 days of onset was very rare, but you can see the highlighted region in the top panel. That's the caudate in the globus pallidus, so that's within the basal ganglia. And the 6 months later, even though the patient still had symptoms, the MRI was normal. So the basal ganglia, as you all know, exerts inhibitory influences on motors and behavior, and motor systems and behavior systems. And we think that inflammation and autoantibodies and injury causes disinhibition, thus disruption of the normal role of the basal ganglia in movements, mood, emotion, behavior, procedural learning, and cognition. And Dr. Pittenger will talk to you about the autoantibodies, which target the cholinergic interneurons within the basal ganglia. Here are some of the neurologic findings we see in our patients. Positive glabellar tap, which is also seen in Parkinson's, failure to sustain tetany, so including the milkmaid grip, darting tongue, tongue fasciculations, and these very subtle choreiform movements of the fingers. If the patient has frank chorea, obviously we call it Sydenham's chorea, but these are more subtle findings. Some of our patients have hung up reflexes, overflow dystonia, and new onset clumsy movements. So we currently follow about 220 patients that meet strict criteria for PANS. And as you can see, the patients in the left panel, the patients have a lot of comorbid symptoms. So even though only 2 comorbid symptoms are required for the criteria, most of our patients have 5 or 6 comorbid symptoms. And again, they all start abruptly. Our patients also have a lot of sleep issues, including this condition called REM motor disinhibition, which is movements during REM sleep, which is highly unusual in children, and it is a predictor of Parkinson's disease in adults. And there's now 2 studies which have found REM motor disinhibition. So the one at the NIH, which showed it in kids with new onset PANDAS, and then our study, which looked at kids with chronic PANS symptoms. And just like Parkinson's patients, our patients also develop dysgraphia. As you can see in the panel on the left, the patient has normal writing. In the panel on the right was during a PANS episode. And typically this writing goes back to normal after the episode is over. And this slide actually came from Dr. Sweeto. So in addition to the motor findings, we also see a lot of immune findings in our patients. So about 14% of our patients have leukopenia or lymphopenia. And many of our patients, I think is about 60% total, have at least one non-specific autoantibody based on the panel we run, which is ANA, antihistone, antithyroid globulin antibody, and thyroid peroxidase antibody. And these are, you know, sort of among the most common autoantibodies in patients in general. So we still have to show whether or not this is more prevalent than controls, but generally reported the rate of ANA in healthy kids is 10%, and in our cohort it's 26%. We also see hypoferritinemia and iron deficiency anemia in our patients with PANS. So about 27% of our patients have low ferritin levels. And we're not sure if that's from gut inflammation or what else is trapping iron. But among the kids with hypoferritinemia, compared to patients with normal ferritin, the patients with low ferritins had worse global impairment, more comorbid inflammatory diseases, and exhibited a chronic course of PANS. And the estimated prevalence of iron deficiency anemia in our cohort was 3 to 8%, which is higher than the age and sex matched US population. Our patients also have low C4, about 34%. We recently did a C4 gene study, which did not indicate that this low C4 that we're seeing is driven by a low gene copy number. The Swedish group at the Karolinska also found low C4 in their patient population. 11% of our patients also have a low C3, and 75% have an elevated C4 ANA, and the C4A correlated with global impairment scores. And as many of you already know, complement activation is associated with immune activation and psychiatric disease. In particular, recently it's been shown that it's associated with severe COVID and neuropsychiatric complications from COVID. So some of the dermatologic things we see prominent onychodermal bands. So these are normal bands that everybody has, but in our patients, it tends to be redder and thicker and more prominent. Typically, this is a faint band that you typically don't see. We also see a lot of periungual redness, livedo reticularis, pallidal petechiae, and to go along with some of these subtle signs of inflammation of blood vessels, we also see a high von Willebrand's factor antigen in 12% of our cases and high D-dimer, but only in 5% of our patients. So it's not surprising that we see concurrent autoimmune diseases develop in our patients, considering the large epidemiologic studies, which show a lot of overlap between OCD and autoimmune diseases, and also eating disorders and autoimmune diseases. So in our cohort, the average age of onset is somewhere between 7 and 9. And about 20% of our cohort now currently has an autoimmune disease. And as you can see, it develops over time, with around, by age 15, about 20%, almost 20% of patients will have developed another autoimmune disease. And for this data, we've excluded arthritis. I'll show you that on the next slide. The most common autoimmune diseases we see in our patients are thyroiditis, psoriasis, celiac disease, Behçet's, and lupus. And again, these autoimmune diseases typically develop after the onset of PANS. We also see that approximately 30% of our cohort develops some form of juvenile arthritis over time. So again, the average age of onset is around 7 or 9. Most of our patients develop the arthritis, or at least appreciated to have arthritis after PANS onset. So the blue line represents the time to pain, and the red line is time to diagnosis of the arthritis. We use imaging in our patient cohort to diagnose arthritis because these patients' psychiatric symptoms often overshadow the symptoms of pain and stiffness. The other challenge we see with the type of arthritis that these kids are getting is that it's typically a dry arthritis, so you don't appreciate swelling on the clinical exam. This is common also in psoriatic arthritis. Our patients also have inflammatory back pain, and many end up meeting criteria for spondyloarthritis. Another feature of the arthritis we see is enthesitis, so they're getting insertion point inflammation where tendons and ligaments insert onto bones. And about 30% of the ultrasounds we obtain on these patients indicate that these patients also have capsular thickening, which is a novel finding in pediatrics. It's never been reported, but it may go along with psoriasis. And psoriasis is also an inflammatory disease that has a connection to group A strep. So this is our overall model of what we think is that, patients get these very common infections, but it's on the background of having some type of immune predisposition. So I'll share with you our HLA and KIR data. Shama is going to share with you some of the innate immune system issues that may be involved. Dr. Pittenger is going to share with you the autoantibodies, which is the adaptive immune system. There's probably a component of the microbiome involved, as has been shown with lots of autoimmune diseases. And also some early studies in PANS suggest that there may be a microbiome component. And then I mentioned some complement, that the complement struck. So basically the combination of the immune predisposition and an infection sort of triggering it leads to a systemic inflammatory response, and we think also involves vascular inflammation and complement activation. And that is what leads to both the PANS-like illness and then as well as arthritis and other autoimmune diseases. So this is sort of our early HLA data where we saw 3 genotypes or HLA-B types that were overrepresented in PANS compared to healthy controls, and those 3 shared an epitope called BW4, and we saw a polymorphism in position 80, which was highly associated with PANS. And this HLA-BW4 is the KIR binding site. So KIR is the killer inhibitory receptor on natural killer cells. And polymorphisms in position 80 refine the binding of the KIR receptor. So we next looked at KIR, since HLA-BW4 and KIR 3DL1 co-evolved. And we also found a polymorphism in KIR associated with PANS. So I'll quickly just show you some of the data here. So these were the original odds ratios we found for B55, B38, and B52. That was our initial 50 cases. But when we expanded to 150, the B38 and the B52 were still positive, but the B55 was no longer significant. And B27 was, which is not surprising, seeing that we see spondyloarthritis and other forms of arthritis connected to B27. And then here's our amino acid level analysis of the BW4. And as you can see, the position 80 is strongly associated with PANS. And it's the combination of having the KIR3DL1 002 genotype with the BW4 confers a risk ratio of about 1.84. And this is also an important gene set that predisposes to multiple sclerosis and myasthenia gravis, which are 2 other neuroimmunologic diseases. So not too unlike what we're seeing with PANS. So just wanted to acknowledge the clinical team in the IBH clinic. All of our patients are categorized as having PANS or not PANS by Margot Tienemann. And we have 2 other psychiatrists that work in our clinic, as well as other clinicians. And then our clinical research team who helps collect all the specimens on our patients and share with people like Tyler and Chris Pinniger and Shama. And then our basic science team, which is largely led by Betsy Mellons, Dave Lewis, Holden Maker, and others, PJ Oates, and our new imaging team, as well as external collaborators. So why don't I stop sharing and hand it over to you, Chris? Thank you so much for joining.
Thank you, Jenny, for that fabulous overview. It's really a pleasure to be here with you today. So Jenny's laid the background of PANS and PANDAS really nicely. I want to focus in on some recent results from my group that are focused on one particular aspect of the pathological processes that she's described, and that's the pathogenic antibodies that are seen in PANS and PANDAS. The model is that a background predisposition in combination with a streptococcal or other infection leads to the activation of the immune system, and one key component of that is the adaptive immune system and the production of autoantibodies, and that's where we have focused. I will just give my disclosures here before going on. None of them are relevant to the work I'm going to be discussing today. All right. So Jenny's talked about PANS, pediatric acute onset neuropsychiatric syndrome. We've focused in these studies on a subset of PANS, an older diagnosis, PANDAS, pediatric autoimmune neuropsychiatric syndrome associated with streptococcus, familiar to many in this audience, I know. So PANDAS is very similar to PANS. I think most people would call it a subset of PANS these days, even though the clinical criteria for the formal diagnosis are slightly different. But it specifically requires the inciting infection be streptococcus. And the reason that we focused on that is because we wanted, if we're looking for the antibodies, we wanted to find as uniform a population of antibodies as possible. So we started with as uniform a diagnosis as we could. And we did this in collaboration initially with Sue Sweeto at the NIH, and more recently in collaboration with Jenny, with Kyle Williams at MGH and others. So these are this is characteristics of PANDAS here. As with PANS and in the data that Jenny's reviewed, there's substantial evidence to focus our attention on the basal ganglia. And what we see here is a neuroimaging study where a patient with PANDAS had edema and evidence of inflammation here in the caudate and putamen. So that's where we focused our efforts. And in looking for the antibodies that might be causative for pandas, rather than looking for the molecules that they stick to, which is what most previous studies had done, we took a slightly different approach, drawing on expertise in my laboratory on the organization of the basal ganglia, and in particular the different cell types that make it up. We focused on not what molecules do the antibodies stick to, but what cells do they stick to. So the basal ganglia, shown here schematically in a horizontal section through the brain, consists of the caudate and putamen. And if you zoom in on the caudate and putamen, they consist of a large number of different cell types. The spiny projection neurons are the majority cells, but there's all of these different kinds of interneurons, which have critical roles that we're just beginning to understand in sculpting and coordinating the overall firing patterns of the cells in the basal ganglia, and thus their output. And so we asked, do the antibodies, this is an antibody shown on the right, do the antibodies in pandas stick to a particular type of interneuron? So we did this mostly in mice. That's just because that's a convenient way to get brain tissue to see what the antibodies stick to. That we have a few experiments from post-mortem tissue from humans, which I'll show you. But in mice, we're able to use a suite of molecular tools, which I'm not going to describe in details today, to identify different cells in the basal ganglia so that we can see where antibodies are sticking. What's just shown here is looking at two different types of spiny projection neurons. The point is merely that we can identify them separately from one another, even though without staining, they look exactly the same. But we can identify them, and we can do that with all of these different types of interneurons. The type of interneuron that we've ended up focusing on, because that's where the data led us, is shown here. It's the cholinergic interneuron, sometimes called a CIN, cholinergic interneuron, sometimes called a TAN, meaning a tonically active neuron, because these cells are in a living animal are active, regularly active. So these are the largest neurons in the brain. I'm sorry, not the largest in the brain, but the largest in the basal ganglia. Now, this is the cell body, and then they have these very extensive dendritic and axonal arbors. They're integrating neurons. They spread over large portions of the basal ganglia, and these are the neurons we've ended up focusing on. And the reason that we've ended up focusing on them is because that's where the data led us. So what we did was we took slices of brain, and we stained for these cholinergic interneurons, shown here in red. And then we incubated those slices with antibodies from children with pandas, taken originally from Susuito's lab at NIMH. And we looked at where the antibodies stick and counted how much they stick to cholinergic interneurons, which you can see from the overlap of the red and the green signal shown here in yellow. And when we do that across a cohort, 3 different cohorts, totaling 27 patients with pandas and 23 matched controlled children with no psychiatric disorder, we see that a large fraction of the patients with pandas have elevated binding to the cholinergic interneurons compared to the controls. Now, it's a little noisy. This is typical in this kind of work. Here's an outlier control that appears to have binding, and there's some individuals with pandas who have no detectable binding. But as a group, there's an elevation in the binding of pandas-related antibodies to cholinergic interneurons. This on the right is just a negative control showing that the number of cholinergic neurons is the same, and the staining for acetylcholine is the same. So this is a key insight that focused our attention on these interneurons. And we've actually seen this now in several different cohorts of patients and using a couple different experimental techniques. Over time, I've come to believe that this is a robust finding. All right. So we replicated that. The data that I showed you were for the binding of human antibodies taken from patients to mouse brain. But we see the same thing when we look at the binding of these antibodies to human brain slices. So these are now slices through the human basal ganglia taken from postmortem tissue. And you can see that the pandas children, every dot here is serum from a different child, show elevated binding relative to control children in 2 different parts of the basal ganglia, the caudate and the putamen. Again, specifically to these cholinergic interneurons shown by the red immunostaining here. All right, so we have binding to these neurons. Does it have anything to do with symptoms? Well, we were fortunate to have a group of 11 children from a study done by Susuito together with my colleagues Kyle Williams and Jim Leckman, where they had treated these children with IVIG. And we selected children who responded to IVIG, figuring that meant they were more likely to have antibody-mediated pathology. And in those children, shown here on the left, we see the binding to cholinergic interneurons shown for all 11 children on the left. This is 2 different cohorts that we tested at 2 different times. That's what the red and the orange are, but they're equivalent. And we see that, so this is binding on the left. This is this binding to the cholinergic interneurons from the same children after they responded, after their symptoms improved with IVIG treatment. Every single child had reduced binding. And when we correlated the change in binding to the cholinergic interneurons, shown on the y-axis here, with the change in symptoms, we saw that children who had a greater change in symptoms, meaning to farther to the right, also had a greater change of IgG intensity. And this was an extraordinarily tight correlation. I love it when data give me correlations that I don't even need to run statistics to know that there's a real effect there. We ran a bunch of controls. We've looked at binding to other kinds of interneurons to make sure this effect is specific. And I'm not sharing all of those negative controls, but they all pointed to the conclusion that this is a specific effect. The data were published last year in the American Journal of Psychiatry, if you want to go look up all of the various controls that we did. Okay, so binding to cholinergic interneurons isn't a completely random finding. Actually, these are a very interesting population of interneuron that we in my lab and others were already focused on as a potential source of pathology in OCD, Tourette syndrome, and PANDAS. And the original reason for that is shown here. This is published work from my colleague Flora Vaccarino at Yale. And what she and her colleagues did in a series of papers, shown to the right here, is they looked at interneurons in the basal ganglia. These are cholinergic interneurons. And they looked at how many there were and how densely they were packed in the basal ganglia in humans who died with Tourette syndrome, not of Tourette syndrome, but with Tourette syndrome. And in every case, they found a reduction in the density of cholinergic interneurons in the basal ganglia. So there's already independent reason to believe that a dysfunction of these interneurons may be associated with disorders in this space, OCD, Tourette's, PANDAS, these overlapping and frequently comorbid disorders. And then in work inspired by these postmortem findings, we in my lab developed a technique to go in and disrupt, actually ablate these neurons in the mouse brain to see what's the functional consequence. This is the kind of causal experiment that you can do in a mouse system that you can't do in human studies. So we went in, and what's shown here is these interneurons, the cholinergic interneurons are shown in white. Our viral tool that we used to focus our ablation solely on those interneurons is shown in red, and all the other neurons are shown in green. So you can see these white and red staining neurons. Those are the cholinergic interneurons. And then when we ablate them, they're gone. You can see there's a few cholinergic interneurons left, none of which are expressing our viral ablation tool. And over here on the far right, we've quantified the number of cholinergic interneurons. This just shows that the system worked, that we've ablated the interneurons, we've removed them from the system. And lo and behold, when we do that, we get twitchy mice. We showed that in a couple different ways. One way here was so at baseline, these mice don't twitch. They look pretty normal. But if we startle them, if we present them with repeated startling stimulus, and then look at how that affects their behavior, we see elevated stereotypy and elevated grooming. That's shown here at baseline. Their grooming and their stereotypy is perfectly normal, but it's elevated when we present them with this repeated stressful, startling stimulus and remains elevated in the period afterwards. Again, we've shown this in a couple different ways, but this tells us that an ablation of those interneurons dysregulates the system in a way that can predispose to repetitive behaviors. Now, these aren't compulsions, these aren't tics, they are, grooming and repetitive behavior in a mouse. But it confirms that these interneurons in the basal ganglia are doing something very important. So they're a logical place to look for pathology in pandas. So finally, if we know that the antibodies bind to those interneurons, and we know that that binding improves with treatment, and we know that dysfunction of these interneurons can lead to repetitive behaviors, that is, maybe causally related to the development of symptomatology, what is the antibody binding actually doing to those interneurons? So here again, we turn to our mouse brain binding system. And we here again in red, you see cholinergic interneurons. What we did now is we incubated slices of mouse brain with the serum from children with pandas. And then we stained for a molecule called phosphoriboprotein S6. I'm not going to go into the molecular details, but the phosphorylation of this protein, the amount of staining that we see for phosphoriboprotein S6 is a measure of the activity of the neurons. It tells us how often they're firing. Remember, I said that these cholinergic interneurons fire quite regularly. They're called tonically active neurons. If they fire less, then the concentration of phosphor-PS6 will go down. And that's how we measured how the antibodies are affecting these interneurons. And here's what we saw. So if we incubate slices of brain with control serum, serum containing antibodies from children who do not have any psychiatric disorder. We do see phospho-RPS6 in the cholinergic interneurons overlapping in red. We quantify them, and this is what we get. When we do exactly the same experiment in parallel, using in this time serum from children with PANDAS, we see a systematic reduction in the activity of those interneurons. If we then incubate the slices of mouse brain with serum from the same children after they had recovered, after IgG treatment, we see that the activity of those interneurons has returned to normal. This reduction, this ability of the serum to reduce their activity has been lost in the post-treatment serum. And finally, if we take the pre-treatment serum and remove the IgG from it, but leave everything else, inflammatory mediators, cytokines, other protein, we see no effect. So it appears to be IgG specifically from serum when the children are sick that binds to these cells and reduces their activity. And that might lead to the same downstream consequences seen in Tourette syndrome when those cells are simply missing, or in the mice when we depleted them and saw repetitive behaviors. So again, adding to the idea that dysfunction, antibody-mediated dysfunction in these particular cells may be a causal contributor to PANDAS. And that's the model that we're continuing to pursue. One last piece of data to show you, everything that I've shown you thus far has been using serum from children with PANDAS from Sue Swedo and her group at the National Institute of Mental Health. Collaborating with Jenny Frankovich, we've replicated all of this work using antibodies from children with PANDAS characterized at her clinic, and we see the same result. We see an elevation of IgG binding to cholinergic interneurons, and we see that when these children improve, some of them with treatment, some of them spontaneously, the large majority of them have a reduction in that binding, adding to the idea that this may be a general mechanism, not unique to just the patients treated by Dr. Swedo. And we are going on working with Jenny to look not just at PANDAS, but at PANS more generally to see if this is generalizable. So in summary, our data have focused our attention on these cholinergic interneurons of the striatum. These interneurons are reduced in number in patients with Tourette syndrome. And when we ablate them in a mouse, we get repetitive behaviors that might be recapitulating part of the symptomatology seen in OCD, Tourette's, and PANDAS. Finally, in the data that I've shown you today, I've shown that antibodies from PANDAS stick to these interneurons, that binding correlates with symptoms, gets better when the kids get better, and it affects their firing pattern, which might lead to the same brain circuitry abnormalities as we get after depletion. So I'm grateful to my collaborators and members of my lab, who the key ones are listed here. Most of the data that I've shown you was produced by John Shu, a very talented staff scientist in the lab. I'm grateful to our funders, including Jenny, who has supported this work, Sue Swedo, and the National Institute of Mental Health, and others. Thank you all for your attention, and I look forward to discussion at the end. I'll stop sharing now, and I believe we move on to Shama Rahman to share some of her work.
Thanks, Chris.
Thank you. Let me share my screen. Okay. Thank you, Chris, for the introduction. I'm Shama Rahman, and I'm a postdoc in Betsy Millen's lab, and we are looking at monocyte and their subsets in PANS patients. So why monocytes? Because a few years ago, Jenny and her team had reported that about 47% of acute onset PANS patients had monocytosis during flare. Monocytosis is an increase in the number of monocytes in the circulation. And there have been previous studies on early onset OCD patients that have shown that there is involvement of pro-inflammatory monocytes in the pathogenesis of these early onset OCD. So in that line of thinking, we wanted to investigate what is the role of these monocytes in PANS. So brief background on human monocytes. These are part of the innate immune system, and they're made in the bone marrow, and then they come out of the bone marrow where they circulate in the blood for a few days, and then they can either migrate to a tissue or they eventually die. If they do migrate to a tissue, they either transform into a macrophage, or they can also generate dendritic cells. In the tissue, they can either, they have various functions, and it can lead to inflammation if that is not properly regulated. So in our lab, we identify human monocytes in the blood by using flow cytometry. So here is a very typical gating strategy that we use to identify human monocytes in the blood. So we start with cells in the blood, and then we identify cells that are single. From there, we identify live cells, and then we eventually get rid of T cells, B cells, NK cells, and other non-monocytic cells from our analysis, and focus on the total monocytes. In humans, there are three major subsets of monocytes. You have the classical monocytes that express high levels of CD14 on their surface. And then you have the non-classical monocytes that do not express the CD14 or have less of that and highly express CD16. And then you have cells that are intermediate, which express both CD14 and CD16. And they have various functions in human, depending on where they are and the type of cytokines they are releasing. So our focus, as I mentioned, was to understand the monocytosis that these PANS patients had. So we got a cohort of PANS patients from Jenny, where we started to investigate the monocyte population in the PANS patients when they had an active disease versus when they were in an improved state. And then we also wanted to compare them with age and sex matched healthy controls. So here you can see that almost all the PANS patients that had an active PANS flare had an increased number of monocytes in their circulation. And for most of these patients, the number of monocytes went down when they entered an improved state. Now, this is the total monocyte count. We don't know from this graph, at least, which monocyte subset is contributing to this increased monocytosis. So we wanted to,
and I just wanted to put in there that those those 3 kids that had higher monocytes, despite their PANS being improved, actually went on to develop other autoimmune conditions. So they were interesting outliers. Thanks, Shama, for letting me interrupt you.
yeah, no worries. Yeah, so we wanted to further investigate what particular subsets were contributing to this monocytosis, so that it gives us an idea of what the roles of these monocytes are in these patients. So this is a very complicated diagram of where these monocytes and other cell types are coming from and how they transform into other subsets. I want to focus your attention to this precursor over here that eventually leads to different monocyte and monocyte-like cells. So you have your inflammatory monocytes and your resident monocytes that can eventually go inside a tissue and become a macrophage or a dendritic cells that in turn would affect the adaptive immune system and would have inflammatory responses. So I wanted to see if there are subsets that are like this in our patients and how they are changing with the disease state. So firstly, we looked at pro-inflammatory monocytes. Here I am showing you one of the pro-inflammatory monocyte subset that expresses these markers that are associated with macrophage polarization. So these monocytes will eventually become macrophages inside a tissue. So you can see that when a patient has active disease, these pro-inflammatory macrophages are elevated in these patients. And all of these patients, when they are in an improved state, the level of these cells go down. We also looked at pro-inflammatory monocyte-derived dendritic cells. Dendritic cells are another type of cells that have that are involved in inflammatory responses in patients, and some of these dendritic cells can derive from monocytes. And we looked at one of those subsets and found that almost all PANS patients had an elevated level of pro-inflammatory monocyte-derived dendritic cells, and then and they eventually go down when they are in improved state. And as Jenny had mentioned, we had patients where we see still elevated level of these monocyte-derived dendritic cells, and these are patients that eventually have chronic static PANS. We compared them with non-PANS related disorders, disorders like asthma, which are chronic, and brain diseases. We don't see these effects in other non-PANS related diseases. So overall, there is an increase in pro-inflammatory monocytes and monocyte-derived dendritic cells when the patients are in flare, and then when they eventually enter remission, these cells go down. And on the other hand, I also looked at anti-inflammatory monocytes. These are monocytes that help in reducing the inflammation. And what we found is that in patients, as they enter improved state, the level of these anti-inflammatory monocytes start increasing in their circulation, which is very interesting because during flare, you get more pro-inflammatory cells. And then when the patients are improving, you get more anti-inflammatory cells in their circulation. Now, all of these cells that we looked at were in the circulation. And as I said, monocytes have the capacity to traffic to different organs. Now, our next question was, okay, we have all these pro-inflammatory and anti-inflammatory cells, but do these cells can they also traffic to different tissues? Our tissue of interest is brain, because the PANS, as you have seen in the previous talks, that there are changes in the brain in this patient. So I wanted to study a subset of monocyte that would have the capacity to enter the brain. And in doing so, to do so, I had to build a panel that would include proteins and cytokines that actually help monocytes to cross the blood-brain barrier and enter the brain. And I picked up 4 molecules that are of interest and that have been shown previously that help monocytes cross the blood-brain barrier and enter the brain, and have been implicated in many neuroinflammatory diseases. So these 4 proteins are CCR2, CX3CR1, VLA4, and CD166. And what I saw is that this first subset that I looked at was a pro-inflammatory migratory monocyte. These monocytes have been picked up in inflammatory bowel disease in a previous study, and I wanted to see if I can find a similar pro-inflammatory migratory monocyte in our PANS patients, and I do. So you can see that healthy patients don't have a lot of these cells, but in PANS patients with active disease, there is a tremendous increase in the prevalence of these cells in the circulation. And then they go down when the patients are improved. And on the right panel, you can see this is the brain homing monocytes. So these cells express all the 4 molecules that are needed for the monocyte to cross the blood-brain barrier. And our patients, PANS patients, when they are in a flare state, almost don't have these cells in their circulation. But when they are improved, there is an increase in the amount of these cells in the circulation, which is impressive. You don't see that effect in a non-PANS related disorder. You don't see them in the circulation of those disease. And this is significantly reduced from healthy controls. So there is a change in these brain homing monocytes in the circulation when the patient is in flare versus when the patient is improved. Now, since these are brain homing cells, they should be going to the brain. So can we pick these cells up in the brain? And to do so, we started gathering cerebrospinal fluid from different PANS patients. We had CSF from patients who had acute onset PANS, and we also got some CSF from chronic static PANS. What I'm showing in these representative figures are the flow cytometric analysis to identify these brain homing monocytes in the CSF. And what we saw is that patients who had acute onset PANS had these brain homing monocytes in their CSF, whereas patients with chronic static PANS did not show these cells in their fluid, which is interesting because we do see a shift in there is less of these cells in the circulation when the patient is in flare, and then there is more of these cells when the patient is in improved, which could mean that the cells that were in the circulation has already started moving into the CSF, and by the time this patient has improved, there might be more in the circulation. And here is an average of all the, here is a number of all the patients that we have looked at. And you can see that all new onset patients have relatively higher number of the brain homing cells in their CSF, whereas the chronic static patients do not have these cells. So whatever these cells are doing, there is a distinct difference in their prevalence in the CSF between an acute onset PANS patient versus a chronic static PANS patient. Now we are trying to understand what does this cell do? Like what could they be contributing to? If they are moving into the brain, are they doing some changes, or are they being pro-inflammatory, or are they anti-inflammatory? So currently this is a work in progress, but we do have some idea from our pilot studies. So one of our working with our collaborator on a mouse model of peripheral inflammation, we found that the CX3CR1 positive cells were actually anti-inflammatory and neuroprotective in the mice, and helped them increase their epileptic response. So it reduced their response when these cells were in the brain versus when these cells were not. And then in our pilot single cell RNA sequencing study, we studied on 1 PANS samples, and we found that these cells could have an immunosuppressive function based on some transcripts that we have picked up in these cells that were elevated in the PANS patient, in the sample when the patient was improved versus when the patient was in flare. We are currently processing more samples, and once they are sequenced, we would be able to understand their function even better. So with that, I would like to acknowledge Betsy and our lab mates, and Jenny and her team, and our collaborators who have helped us with giving us non-PANS related samples and help us understand the brain homing cells in mouse model, and our funding sources. Thank you. And I think Tyler is going to talk next. Thanks.
Thank you, Shama, for that introduction, and thank you all for the opportunity to speak today about our work, which I see very much as complementary to Dr. Pittenger's. While he is focused on the neurological side of things and brain side of things, we're looking at the autoantibody response in PANS and trying to understand how the immune response is changed in PANS and pandas. So in terms of the pathogenesis of PANS and pandas, much of the evidence that we use to understand this illness currently comes from the related post-streptococcal illnesses, including Sydenham's chorea, which is thought to have a similar pathogenesis. And as Dr. Frankovich and Dr. Pittenger already went through the implication of the basal ganglia and inflammation in that region of the brain, Sydenham's chorea has been found to have inflammation there, as well as autoantigens that are thought to arise from the inflammation in the tissue there. The target autoantibodies that have been identified so far include lysoganglioside, tubulin, and both D1 and D2 dopamine receptors. These findings have been extended to PANS and pandas. However, the studies to date have been somewhat mixed in terms of the prevalence of these antibodies. And so this is something we're very interested in, and I'll talk more in detail about that. But from our standpoint, we're a lab that specializes in studying traditional autoantibodies from autoimmune diseases. And so we have methods developed for that that we'd like to apply to PANS and pandas to understand how the immune response is changed or how it's unchanged in that setting. And then also wanted to look at antibodies against the GPCRs that have been implicated, as well as a whole host of other GPCRs that are very important, which I'll talk about in more detail. And the goals for doing this are for us to understand a little bit more the diagnostic capability of looking at these specific autoantigens to see if we can understand if there are multiple clinical entities, and that would help us design targeted treatments and then potentially develop novel treatments if we're able to discover novel targets. And so the method that we use is focused more on a reductionist-based approach, as Dr. Pittenger was talking about, where in our case, we have autoantigens coupled to fluorescent beads of unique bead IDs. We're able to measure up to 500 different protein antigen targets and look at autoantibodies that bind to those simultaneously and decouple the signal for each one using secondary antibodies to detect the IgG bound to the antigens. We can get a readout for how much antibody is bound to any individual autoantigen. And so when we measure traditional autoantigens, we look at a variety of illnesses, including rheumatoid arthritis, systemic sclerosis, thyroiditis, lupus, as well as GI and endocrine autoimmune disorders, and the targets that are commonly associated with those disorders. And so what we've done is taken serum from PANS patients as well as healthy controls and compared the relative abundance of these antibodies across these populations. And so on the left, you see patients, and then on the right, healthy controls next to that. And the red represents samples that are greater than 5 times the standard deviation of the healthy control signal background, and then over an MFI of 3,000. So it gives us an indication that there's an antibody present there. And as you can see, when we look at a large cohort, we don't see particular differences in the autoantibodies that are prevalent in response to these traditional autoantigens. On the right are shown some controls to make sure our assay is working, as well as negative controls on the far right. And these results contrast a prior study. Actually, when we had submitted our original abstract, we had done a smaller group of samples, and it seemed like there was a generally increased prevalence of autoantibodies in PANS patients. However, those samples were selected as patients with in elevated levels of autoantibodies. When we later went back and included patients with high CY-BOX scores, as well as more healthy controls, there was no clear difference in autoantibodies against these traditional autoantigens. So we're currently getting a larger cohort of samples so we can have much more statistical power to determine whether or not there is any difference in response to these antigens. The other thing I wanted to talk about, something we're really excited about, looking at autoantibodies that bind to GPCRs. You may or may not have heard of GPCRs, but they are the most diverse set of eukaryotic membrane receptors and represent 30% roughly of drug targets. So they're very important pharmacologically. However, a major limitation to working with these proteins that's existed so far has been that when GPRs are pulled out of cell membranes, they're almost like a tent having the tent poles removed, where the structural stability is totally lost. And so it's hard to maintain conformational epitopes on these proteins. And that's really important for GPCR function, because the conformation of the function is actually, or the conformation of the peptide is actually what dictates the function and the signaling. For these reasons, we've collaborated with Dr. Brian Kabilka's lab at Stanford, and their lab specializes in studying GPCRs. So we've collaborated with them, one, to get the functional purified proteins, as well as to come up with ways to modify our existing methods to be compatible with GPCRs and preserving their structural integrity. So the modifications that we've had to go through have included, rather than directly linking antigens together, we have found a way to indirectly immobilize the GPCRs without affecting the GPCR structure. We've had to substitute the harsh detergents used traditionally to examine autoantibodies with detergents that are compatible with studying membrane proteins. As well as using several other reagents that we've had to modify so that we can reliably detect these GPCRs held in their appropriate conformation according to crystal structures. And one of the ways we can validate that is that the Kabilka lab developed a nanobody that is able to recognize conformationally specific epitopes. And so the method as we currently have developed it involve immobilizing GPCRs using flag tags to beads with anti-flag antibodies bound on their surfaces. And in this way, we're able to detect the difference in protein conformation when either, in this case, our representative receptor is beta-2 adrenergic receptor. When beta-2 adrenergic receptor does not have a ligand bound, the nanobody I was describing does not bind to that protein. However, when the ligand, in this case, it's a very high affinity ligand, binds to the protein, the conformation of that GPCR then becomes accessible, and the nanobody can bind. This is our way of determining whether or not the function and the conformation of the protein is preserved. And this is in the middle right. It's a panel showing representative data from this. So only when beta-2 is present, immobilized on the flag tag antibody, and in the presence of agonist, are we able to detect the active form of beta-2. And then shown on right, we have the compatibility with human sera. So again, only when the agonist B, represented B there, and beta-2 adrenergic receptor are present do we see the maximal binding, in contrast to when I, the antagonist, is present. And so after many iterations, we were able to develop this assay. And we've gone through now and added many different GPCRs, and we've been going through making this assay repeatable and making it possible to be able to multiplex with many different receptor and ligand pairs. As you can imagine, there are incompatibilities between certain receptors and certain ligands, so there's a lot of optimization that has gone into this. But we're excited to be getting to the point where we can now go through and analyze patient serum. So the next step for us is to obtain a large cohort of pandas and pan serum and screen for reactivity against GPCRs. Through this novel method, we're going to be able to look at conformational specificity of autoantibodies. So agonist bound forms representing active forms of receptors, as well as antagonist bound representing inactive forms. We want to compare this with the traditional membrane or traditional assays that use ELISAs, including those that have used cell membrane extracts, and see what are the relative findings using those traditional methods compared to our novel method. And then finally, we want to validate our work through functional assays, which we're also currently developing. That's all I have. I'll stop sharing here.
That was excellent, Tyler. Thank you so much for presenting. So that concludes our panel for today, and we look forward to answering your questions at our next gathering. Thank you very much, everyone.